Alright, today we're going to talk about nucleic acids. And you can see on the slide here that you have all kinds of things with nucleic acids, and any type of cell has nucleic acids. You can see the different foods that we eat. Hopefully you're getting a little hungry seeing some of the things on the bottom. Even the little kitty here that we know Mr. Grill would like to eat, but we're not going to let him do that. She's full of nucleic acids, as well as the big fat guy sitting behind the computer right now. So let's get going on discussing the last of our macromolecules, the nucleic acids. Alright, these are responsible for information storage. So we want to store genetic information. It's nucleic acids that we're going to use for doing this. And the little Google ad here is pretty neat. I'm assuming they did this on the birth date of the discovery of the double helix. Nucleic acids, and I apologize, you guys are going to hear all the sounds. I at least got them turned off on this computer so you don't hear the echoes, but you're still going to hear all the sounds. So if I'm talking over the top of them, I apologize. Suck it up. Deal with it. Alright, these are the genetic material. So you can see the chromosomes here. Alright, and really all the chromosomes do is they store information. It's the big filing cabinet of your cells. The information is also filed into genes. Alright, and then these are going to be your blueprints or your instructions for how to actually make a protein. And remember, proteins do just about everything. If you haven't gotten that stuck in your head, you're going to want to get that stuck soon. Alright, and so really when we look at the nucleic acids, what we're looking at is we're looking at the DNA and the RNA. Alright, so you have the DNA, which gives the instructions for creation of RNA, and the RNA gives the instructions for the creation of particular protein. Alright, and so each location, each gene, then is going to act as the recipe for one particular type of protein. Just like the analogy I always use is you think of a cookbook. In a cookbook you have hundreds, in some cases thousands, of recipes all in there. Each individual recipe is similar to the gene within the recipe book that we're going to call the DNA, or chromosome in this case. Alright, so you can see the instructions here in the DNA molecule are used to help program the creation of a protein molecule. And Ariel's part of all that too. So, alright, let's keep this going. These also then, therefore, help with transferring information because DNA is really what stores it. RNA is what transfers it. Right? And so we can also look at DNA here in terms of its ability to help transport the necessary information for new cells to be able to exist, as well as to help with being the blueprint for the next generation. So we're really talking two different processes here. The blueprint for making new cells, so we're talking mitosis, and the blueprint for creating cells that will be passed on to the next generation, or the process of meiosis. You can see the creation of many sperm cells there. So just a couple cool little animations here. And you can see the chromosome is tightly compacted here, and then it gets unwound, and you can see the DNA helix. Alright, so when we talk nucleic acids, really there's two big ones. There's the RNA, which we call ribonucleic acid. And RNA, if you look at this graphic right here, I'm going to be fighting the cat the whole time. If you look at this graphic right here, you can see it's only one-sided. So one of the things that makes RNA unique from DNA is it's a one-sided strand. Alright, so we call that a single helix. Alright, then we also have the DNA, which we call deoxyribonucleic acid. Alright, and the DNA is two-sided, so we call it a double helix. Alright, so you can see obviously the RNA then single-sided, DNA has two sides. Alright, when we talk about the structure of these, and that's really what we want to get into because we're talking the macromolecules and all the structural components of the cells, the monomers that make it up, or the individual pieces that make up a nucleic acid, we call those a nucleotide. It's a pretty important piece of information you want to retain for the upcoming test. So when we look at nucleotides, what we're really looking at is there's three parts to every nucleotide, and you learned all this back in basic biology, but we're going to take it a step further. First of all, we have the we call it a nitrogenous base. All right, so you have a base that really is what gives each nucleotide its own unique identity. All right, and it involves a carbon and nitrogen ring, and you'll see some examples of that here in a few minutes. All right, so you can see the nitrogenous base on the DNA molecule right there. Right, and so really, going back to sophomore biology, that's where the A, T, G, C, and U, and I gotta get rid of the cat, so hold on a second. Sorry about that. And then we also have a sugar, and they call it pentyl sugar, and I'm not too concerned about you being familiar with that. But there's really two pentyl sugars that are involved in nucleic acids. There's one called ribose, and there's one called deoxyribose. Ribose is the name of the sugar you'll find in RNA, and remember, if you're trying to remember whether it's a sugar or not, look at the OSE. The name of the sugar in DNA is deoxyribose. Now, it makes sense why we call it RNA and why we call it DNA then. And so I'm going to give you guys a second, and I'll stand or sit here, be quiet a second, see if you can figure out the difference then between ribose, which is found in RNA, and deoxyribose, which is found in DNA. And hopefully what you're able to find is right there. There's an additional hydroxyl group on the second carbon on the RNA molecule, or on the ribose molecule of the RNA. And then there's also a phosphate group. And we talked about sugar phosphate in sophomore biology. So now you have a better idea of what a phosphate group is because of us talking about all of our functional groups. So you can see the phosphate group which I just circled there, and I got circled again. Alright, so based upon this, you can see the little penguin guy is asking about, is this a charged molecule? Well, hopefully you can figure out, yeah, because of the fact that it has a phosphate group attached to it. So some of our different nucleotides, there's really two different types, and we didn't go this far when we talked about DNA before. There's your purines, which include adenine and guanine, and what you'll see with these is that there's actually a double ring structure on it. Both rings contain nitrogen within them. Alright, so adenine and guanine are your two purines. And then you have your pyrimidines, which are just one ring, so a single ring that once again has nitrogen within it. And that includes your cytosine, your thymine, as well as your uracil when you're talking about RNA. Alright, and so if you remember right, A always goes with T in DNA, G always goes with C in DNA, and then when we're talking about RNA, we talked about G always goes with C, and for making RNA from the DNA template, A goes with U. Alright? So when we look at the polymer of a nucleic acid, let's start by looking at the outside. So we're going to section this off right here. And what we're really looking at here is we have a sugar that's bonded to a sugar here bonded to a phosphate group. Once again, a sugar bonded to a phosphate group, a sugar bonded to a phosphate, sugar bonded to a phosphate, all the way down the entire backbone. Well, because we've had different names for the bonds that help to hold together the monomers in each of the macromolecules, we once again have a name for this one, and we're going to call it a phosphodiester bond. So add that to your list of the type of bond you should be able to identify for the purposes of this class. All right, and so you can see a single monomer of DNA here, or of RNA, I guess it could be either one, and then you can see we have our nitrogenous base attached to a sugar, attached to a phosphate. And then the bond that's going to hold this phosphate to the adjacent sugar we call a phosphodiester bond. Same thing going on here, holding this sugar to the adjacent phosphate. Alright, and you can see that highlighted there. And again, and it just keeps happening all the way throughout the strand of the nucleic acid. Alright, so here when we talk about this, what we're looking at is new bases added to the sugar of the previous base. Alright, and the polymer is always going to grow in one direction. I'm not worried about you knowing about 5' and 3' ends of the nucleic acids right now. We'll get into that when we actually get into the structure and a little more specifics with nucleic acids and how they're involved in protein synthesis. So we'll hold off on that for now. But what you can see here is all of your bases are kind of hanging off to the side, and that's pretty important because if you remember back to talking about DNA, the structure of DNA, DNA replication, when we talked about the formation of RNA, so when we talked about transcription, we need to have the ability for some hydrogen bonding to occur between the bases and the template from which that new strand is being made. Right, so the fact that these are exposed like they are is going to be helpful in being able to allow for that hydrogen bonding to be able to occur. Alright, so when we pair these up, we already talked about A always goes with T, C goes with G on the DNA strand. One thing we didn't get into in the sophomore biology is the fact that when you have A and T bonding together, using those hydrogen bonds, you notice there's two hydrogen bonds that connect them together. We only talk about a hydrogen bond. Well, that's because you obviously weren't ready for getting into the chemistry, but now that you guys understand a little bit more about the chemistry behind all this, there's actually going to be two hydrogen bonds that hold the A's and T's together in a nucleic acid. And there's going to actually be three of them that hold together the guanine to the cytosine. And in each case here, we have a purine getting attached to a pyrimidine. So here's your purine, and here's your pyrimidine. Purine, pyrimidine. How do pyrimidine, purine. I said that backwards again. Pyrimidine, purine. I'm just completely messing this up. But you get my point. What you should be looking for is here I have on this nucleic acid, or excuse me, this nucleotide, wow, I am struggling here tonight. Anyways, on this particular nitrogenous base, you have two rings. On this one, we only have one. The ones that have two rings are your purines. The ones that have one ring are your pyrimidines. So thymine and cytosine were your pyrimidines. Here's your purines. If I messed that up, I apologize, and I'll get back to you on Tuesday with that. Alright, so when we look at the DNA molecule, it's twisted into a double helix. Alright, and you can see in terms of relative size, it is extremely small, 1 nanometer. Alright, and you're always going to see the A going with T, C going with G. Alright, and it's hydrogen bonds that are going to help to hold those bases together. Why is that important? Well, we talked about in the past, you needed this to be able to unzip for DNA replication to occur. You need this to be able to unzip in order for transcription to occur so that you can make RNA that can then be sent to the ribosome and used to be able to form a protein. So there's two pretty important reasons why we want these bases to be held together by a hydrogen bond. Makes it a lot easier for us to be able to unzip and unwind the DNA. So let's talk about replication just very briefly. And when we talk about replication, really it's nothing more than the copying of the DNA. Right, and what you're going to see is the two strands of the DNA that get made in replication are going to be identical to one another in the very end. Right, DNA is what we call a complementary structure, so that means the one side is complementary to the other side. We already talked about the fact that the bases match up within one another. That's what we mean by complementarity. All right, so if we have one side, then we know what we need to build the other. All right, so as long as one of the sides exists, we can have something, primarily a protein, that's going to go through and help to be able to build the other side. All right, when do we need to copy this stuff? Well, we need to be able to copy it for multiple reasons. All right, so one of the reasons is cell reproduction. Your cells don't last forever. We got to be able to replace cells that have died. We need to be able to make new cells for growth purposes. Multiple reasons why we need to be able to make new cells. So we need the DNA to be copied whenever we're going to have one cell split and become two cells. Each of those sister cells needs to have a full complement of the DNA. So we need to be able to copy the DNA prior to mitosis. And we'll get into the cell cycle a little bit later on in the semester. All right, so you can see a cell here undergoing cell division pretty much near the end. And we looked at these. I'm assuming this is an onion cell, and they used a little bit different staining than what we've used in the past. And you can see it clearly, the DNA splitting apart. All right, you also are going to need copies of the DNA to be made when you're making new sperm and egg cells. All right, so the process of meiosis, so making your reproductive cells. And so you can see sperm cells that have reached an egg cell and are trying to cause fertilization to occur. When we look at the history behind DNA, these are two guys you've probably heard of, James Watson and Francis Crick. These are the guys that are given credit for determining that DNA is found in a double helical structure. And there's a few others, and I'm going to skip through those pretty quickly. All right, so when we look at this hydrogen bonding is going to have a big impact on how structurally stable the DNA molecule is. All right, if we have only 2 hydrogen bonds, it's not going to be held together quite as strongly. If we have 3, it's going to be held together a little bit more strongly. Another way of saying that same thing is, if we want to be able to break this, we're going to need more energy to be able to split apart the guanine and the cytosine than we are to split apart the adenine and the thymine. Well, why is that important? Well, think about some of the biotech procedures where some of the things, when you're getting into DNA technology, some of the things that you're dealing with, you have to increase the temperature in order to be able to replicate this a little bit more quickly. And so we need higher temperatures to be able to make this happen. And I'm not too concerned about this one. This is the one I'm more concerned with. Organisms that are adapted to be able to survive at higher temperatures, well, you don't want their DNA to be able to be split apart that easily. So it's more of an advantage to have a lot more guanine and cytosine than it is to have more adenine and thymine, because it's going to be able to exist at a higher temperature compared to what would happen to the hydrogen bonds between the adenines and thymines. All right, parasites, for some reason, we don't really know why, seem to have a lot more adenine and thymine combos than guanine and cytosine. All right, here's something that's pretty interesting too. ATP. We've talked about ATP briefly. We haven't spent a lot of time on it, but your energy currency for the cells, adenosine triphosphate. Think about what we just talked about with the nucleic acids. We said there was a phosphate, and then we also said something about adenine, and if you look closely, adenine. So these must be pretty closely related. Well, if you look at it, here's adenine, here's your ribose, so we're really talking a nucleotide of RNA. And instead of having one phosphate, I have 2 more. So ATP is a very closely related molecule to the nucleotide for RNA of adenine. Alright, so and this is just going through what I just got done saying. So here's your adenine molecule, or excuse me, your adenine nucleotide, and all we're doing when we're talking about making ATP is adding 2 additional phosphate groups to it. Huh. Good stuff. Alright, so another helical structure. And we'll do this macromolecular review when I come back on Tuesday. And so I just thought this was a perfect slide to be able to end it. That's the basics behind DNA. And just thought this was a cute little graphic to be able to put an end to this video. So I hope this helps. Make sure you guys, if you have any questions, let me know on Tuesday. We'll get it taken care of. And I hope you guys have a great day. Get some good work done today.